Hello, dear students. Have you ever thought about this? We can measure temperature with a thermometer, time with a watch, and even heartbeats with a stethoscope. But how do we measure something invisible like transpiration? Plants are constantly losing water vapor, but we can't see it with our eyes. That's where scientists design smart methods to detect and even measure this hidden process. Today, let's explore the fascinating tools and experiments that make the invisible measurable. Let's begin with the simplest method, the gravimetric method. Here, a well-watered potted plant is weighed at regular time intervals on a balance. As water is lost by transpiration, the plant gradually becomes lighter. If we cover the soil surface with oil or wax to block direct evaporation, then the weight loss is due mainly to transpiration from the leaves. It's just like how fruits or vegetables lose weight and become shriveled when left outside for a few days because water is lost slowly into the air. Now let's look at the most famous device, the potometer. The word poto means drink, and this instrument measures how much water a plant drinks in as it transpires. Here's how it works. A freshly cut leafy twig is fitted into the apparatus filled with water. We introduce a small air bubble into the capillary tube. As transpiration occurs, the plant absorbs water to replace the loss, and this makes the bubble move along the tube. The faster the bubble moves, the faster the rate of transpiration. Isn't it amazing that such a tiny bubble can act like a biological stopwatch for plants? But remember, the potometer has its limits. It doesn't actually measure water lost by transpiration, it measures water uptake. Some of that water might be used in photosynthesis or stored inside plant tissues, so results are only approximate. Also, if the shoot isn't cut underwater or the seal isn't airtight, air may enter and disturb the reading. Even with these challenges, potometers remain one of the most trusted tools for comparing transpiration under different conditions like light, wind, or humidity. Another clever way is using cobalt chloride paper. This special paper is blue when dry but turns pink when it absorbs moisture. When pressed against a leaf surface, the paper gradually changes color, proving that the leaf is releasing water vapor. While this method doesn't give us the exact rate of transpiration, it's a quick, visual demonstration, perfect for classrooms and lab experiments. Now let's compare transpiration from different leaf surfaces. If you cover some leaves with waistline, blocking the stomata, and leave others uncovered, you'll notice that coated leaves lose little or no water, while uncovered ones transpire normally. This experiment clearly shows that stomata are the main exit routes for water loss. It's similar to how applying lotion on your skin prevents water from evaporating, keeping your skin from drying out. Plants use a similar barrier when their stomata are closed. So students, let's connect all that we've learned today. Transpiration may be invisible, but it can be measured. The gravimetric method shows weight loss due to water vapor. The potometer method tracks water uptake using a moving bubble. The cobalt chloride paper method detects water presence with a color change. And comparative experiments with waistline prove that stomata are the main pathways. Isn't it brilliant that with just balances, bubbles, and bits of colored paper, we can measure such an invisible yet vital process? I hope you now appreciate how science uncovers hidden plant functions with creativity and precision. Biology truly is the science of life in all its wonder. Until next time, keep observing the living world. It always has secrets waiting to be discovered.